Our next speaker is Nuno Enrique Franco, and he's a researcher at the University of Porto at this I3S, and he's the president of the EdPlus and vice president of the Portuguese Society for Laboratory Animal Science, has been board member of Animal Research Tomorrow, former Basel Declaration Society, and that's coordinating the Portuguese network of animal welfare bodies. So as you can see, he's a very well prepared person, and we're looking forward to hear from him stress-induced hyperthermia in mice from routine procedures, why it matters. Thank you, Nuno.
Hi, everyone. I hope you're hearing me correctly. Thank you for inviting me. Obrigado por me terem convidado. Gracias por haberme invitado. And now switching fully to English. Oops, almost clicked the wrong button here. So I'll be talking to you about stress-induced hypothermia in mice from routine procedures and why it matters. A very quick overview of this talk. Actually, the title says it all. So I'll first address what is stress-induced hypothermia. I'll then move on to talk about how you can observe this phenomenon in mice from routine procedures, and then I'll wrap it up by stressing why this should matter in terms of animal welfare and experimental design and the quality of the scientific output. Have you ever felt when you're very stressed that you are actually heating up as if you had a fever? Well, actually, you are heating up. This is a common response in most mammals. And although sometimes it's called psychogenic fever, the correct terminology is stress-induced hypothermia. One of the reasons is because it's not actually a fever. So it is characterized by a body temperature rise. In a way, it's a fight or flight response to an acute stressor. In rodents, the temperature may rise 2 to 3 degrees in extreme cases. So the thermogenesis is mediated in the brain by the rostromedullary rapid region and the dorsomedial hypothalamus. I hope that made sense to you, because it doesn't do much in my case, but it does involve the activation of the HPA axis, as in all stress responses, and the thermogenesis is achieved by brown adipose tissue thermogenesis. There is peripheral vasoconstriction, directing the blood circulation to the heart, the muscles, and it is accompanied with moderate tachycardia. It is because of this vasoconstriction in the peripheral regions, it is associated with a temperature drop in the extremities. So while the body is heating up, the tail, the paws, the ears of the animal will be, if it is indeed a stress response, will show a drop in the temperature. The duration and the magnitude of this response is affected by the intensity of the stressor. And even if you have chronic stress rather than acute stress, it might then induce an anticipatory hypothermia and chronically increase the basal body temperature. On your left, you can see in this graph, I'm sorry, changes in abdominal temperature in rats after social defeat. Social defeat is a very strong acute stressor, and you can see how the temperature goes from 37 degrees up to almost 39. But I would like to call your attention that the control groups are sham handled. So rather than undisturbed animals, they picked up the animals. And you can see that actually there is a significant increase in temperature, even though the animals are only being handled, and you can see that the intensity of this response is smaller, but there is a response that is observable just from the handling stress alone. So the measurement of temperature, especially if you're using a thermometer, regardless of it being a rectal thermometer or an infrared thermometer, can itself be stressful and affect the outcome, because you have to contain the animals. Well, one of the possible alternatives could be using radiotelemetry, but then sensors, especially if they are battery operated, might have an impact on animal welfare and physiological parameters that are of interest. There may be variation from sensor position, local information. It does require surgery. It is costly and time consuming. An alternative is the use of much smaller, battery-less, thermosensitive, passive integrated transponder tags, or PIT tags. They are much smaller, as I said. They have only 12 millimeters. They also provide a unique identifier, and you can store the information on how many measurements you take for up to thousands of records. It's much less invasive. It is usually well tolerated, can work for years, and it can, because it has an identifier, and with each reading you can identify which animal has been bred. You can use it in automated assessment. There are some cons. Sorry. It should ideally be implanted under binge 3 to 4 minute anesthesia, even though it's not a surgery. One of the reasons is because it is recommended to add a small drop of surgical glue in the puncture site because it's relatively large. The reading has to be done at very close range, roughly less than 5 centimeters. In some cases, this means removing the animal from the cage, which defeats the purpose of contactless measurement. The tags can move from original position, and in some cases, in our experience, it's roughly in 5% of the cages, the animals have to be re-tagged because they remove either their tags or the tags from their cage mates. And it does require a few days for adaptation to the new object and the healing of the puncture site. Another approach that is growing popularity is infrared thermography. It does have some cons. First is that body surface temperature is not and should not be considered a proxy of core temperature. Perhaps except in the eyes, but the problem is it will only work if you actually pick up the animal and place it in front of a thermal camera, and again, defeating the purpose of contactless assessment. And in response to acute stress, the temperature in the extremities will drop. So this could be sending confusing signals. And the output can be affected by environmental conditions, so the temperature, the humidity of the room, and the distance to the objective. But there are ways of tackle these caveats, which also include the laboriousness of image analysis, as anyone who has done image analysis can testify. One is just do not consider body surface temperature as a proxy to body temperature, but as a parameter in and of itself. Also, the fact that you can see different variations in different regions actually allows to get more information on the thermobiological response. And also, if indeed you observe a body temperature rise in most of the body and a drop in the extremities, you have information on the valence of this aroused state. You know that it has a negative valence, that it is a result from acute stress. Another advantage in the lab is that in animal facilities, there you have highly standardized environmental conditions. And in regard to the laboriousness of image analysis, in the I3S, we developed the software for automated high throughput analysis of thermal images of mice. We have also established that mean body surface temperature is a robust parameter for following temperature variations. This work was mainly carried out by Canon Yendel, and it's much more robust than eye temperature because the animals tend to tilt their eyes, and the angle might shift even though you have the camera at a fixed position. And though the tail can be quite informative, as I said, because of this vasoconstriction, it might send confusing messages, and you'll also have a low inter-observer agreement. And this software that we use, that was developed by Ana Geroz and Paula Guiar, and that was presented in this publication that you can see here, Has as technical features that it is compatible with a universal format, which is the CSV file. What it does is automatically removes the background and just highlights the mice, provides the mean body surface temperature and the median temperature from each mouse, either from each mouse or from the mice as a group. It generates a 3D heat map, as you can see in the bottom left corner of the image. You can also calibrate it, fine-tune it, and rather than analyzing one image at a time, you can analyze hundreds of images at a single time. You just run the software, grab a coffee, and then when you return, you'll have the data. And we test compatibility with at least two manufacturers sorry, of cameras, thermal cameras. And we now have a new improved version that can segment the animal using machine learning algorithms to separate the temperature from the body and from the tail, because the tail is so informative in terms of the valence of the stimulus. So we set out to check whether we could use thermal imaging for measuring hypothermic stress response. We had a number of hypotheses. These have all been pre-registered in the animalstudyregistry.org as a measure to prevent p-hacking and hacking, which is hypothesis after results are known, and is a good practice that I recommend to everyone. And the hypothesis were that infrared thermography would be able to detect stress-induced hypothermia just to a mouse stressor response to handling, that this data would concur with data from the thermal-sensitive pit tags, that mice handled by a tunnel would show a lower stress-induced hypothermia than tail-picked mice, because it has been reported that handling the mice via tunnel is less noxious than picking them by the tail, and that if we use anxiogenic behavioral tests such as the elevated pulse maze, we would see this effect as stress-induced hypothermia. And if it is indeed a result from stress, if you give the animal an anxiolytic, then animals treated with anxiolytic would show a lower response than the vehicle controls. This is roughly the study design. We used two strains of two sexes. It was a randomized block design, because we divided the 68 animals into 3 cohorts. Each cohort was treated as a block, a random factor. All treatments were assigned randomly, as one does. The analysis was carried out by a blinded observer. The mice were handled twice every day for 2 weeks, and we paired the behavioral studies that we carried out with the thermal assessment using infrared thermography and the reading of subcutaneously implanted pit tags. So we decided that it would be best to use both methods rather than just one. And of course, this would also allow us in a way to validate the use of either method. And we provide the animals 1 week to habituate to the tag implantation and to the habituation to the tunnels that were in the home cage. The behavioral tests that we performed were basically collecting video from daily handling. We just picked the animals from the cage. The animals were housed in pairs, placed them in another cage for 2 minutes, and returning them to the original cage. We did this every day for 2 weeks, and after 1 week, after each of the weeks, we did a voluntary interaction test where the handler approaches the bench, and we basically measure how much time the mouse spends in the half of the cage that is closer to the handler, and how many interactions with the handling apparatus, either the hand for the tail-picked mice or the tunnel for the tunnel-picked mice. And at the end of the 2 weeks, we carried out the elevated plus maze, and the parameters, as one knows, is the time spent in the open arms and the number of entries in the open arms. So if the animal is more anxious, more stressed, it will spend more time in the closed arms and perform less exploration in the open arms. So as expected, the tunnel handle mice interacted significantly more with the handling device, but more on that in a minute. We did not find, however, between tail-picked mice and tunnel-handled mice any significant differences in the elevated plasmase. We did find, as expected, of course, differences between diazepam-treated animals who spent more time in the open arm and had a higher number of open arm entries. Every day, we observed that the temperature of the mice would rise within 2 minutes, a mean rise of 0.6 degrees for all strains in all sexes, and no difference between handling days. And this was observed in the subcutaneous temperature measured by reading the pit-tex, and in observing the mean surface temperature using thermography. No differences between, again, tail-picked and tunnel-handled mice, but indeed strain and stress differences. Biopsy mice and males showed higher temperatures in the daily handling. And this was also observed for the two methods, reading of subcutaneously implanted pit-tex and measurement of mean body surface temperature. What we did not anticipate was that the second animal being handled would have a significantly higher temperature than the first animal handled. So handling order had an effect. And we know that the effect is anxiogenic because the animals were warmer in terms of body temperature using both methods, but using infrared thermography, we observed that the tail temperature was lower in the second animal handled. And again, we found testing order to impact body temperature significantly. And we didn't find, so both in the voluntary interaction test and during daily handling. This is for the elevated plasmase, as expected, due to the anxiogenic nature of the test. The temperature rose significantly during the test, and the diazepam-treated animals showed a lower, as you can see in this bottom right corner, a lower temperature rise, and also observed from the beginning of the test. And the testing order did have a significant impact at the beginning, but then it evened out during the test. So we know that now that handling elicits quick onset mild stress-induced hyperthermia. We can observe it by both methods. No differences in stress-induced hyperthermia across the 10 handling days, and no significant temperature differences between tail-picked and tunnel-handled animals. Our hypothesis is that perhaps there were differences, but in the context of the interference of other stresses, removing the animals from the rack, taking them to another room, opening the cage lid, these are stresses that will probably mask small effects from differences in how you handle the animals. The tail temperature drop confirms the negative values of the aroused state identified by hypothermia. And also our data is validated by the EPM performance and the mean body surface temperature differences between vehicle-treated and diazepam-treated animals. So this is very quick.
You need to finish.
I will finish in 2 minutes.
This is exactly what you have.
Okay.
No questions.
Great. Yes, you can send them by email. Thank you. We actually did a second exploratory study. We haven't published this one, to check whether cage change would also elicit hypothermic stress response. And basically, we compared animals that were undisturbed. We did not remove them from the cage, and we monitored them for 4 hours. The same for animals for which we changed the cages, and the animal went to another cage. Animals that went to another cage, but with some odor clues from the previous cage, and animals that we just picked up and returned to the original cage. All disturbed animals raised the temperature quite higher, 1.5 degrees higher than the animals that were undisturbed, but there were no differences between the animals that were just picked up and handled and the animals that were moved to a new cage. And I would like to bring your attention that this stress-induced hyperthermia persisted up to 4 hours. So why it matters? So we showed that we can use both infrared thermography and PIT-X to non-invasively follow temperature variations. The method can allow assessing the impact of acute stresses. And because infrared thermography allows to check the temperature of the tail, it informs on the valence of the aroused state. And you can use the PIT-X and infrared thermography to reduce experimental noise from restrained stress. Mind that the testing order impacts the outcome. So it's quite important to randomize the order by which you carry out any tests, and also that there was no habituation effect. Even after 10 days of handling, the animal still had a stress-induced response to handling. This raises the importance of automating and monitoring animals as undisturbed as possible. And I would like to invite you to know the cost action tea time, which actually focuses on improving biomedical research by automated behavior monitoring in the animal home cage. And we have just basically started this cost action. And we are also working to automate this using Arduino-based
Thank you, Nuno. We need to
sensors.
finish.
Thank you.
Thank you.
Thank you to all offenders for you to listen.
Okay, thank you, Nuno. And this is just one question from me. So what about the time parameter? Because you are calculating and monitoring temperature in different parts of the body of the animal, but I guess this is monitored continuously. So the average temperature is taken in a given moment in time, or is calculated through a different number of minutes?
We actually selected a few time points. Of course, this impacts how many comparisons we have. So it's good to not make that many comparisons, because then you have to correct for multiple comparisons, and you might lose statistical power. For the problem with infrared thermography is that you have to have the cage lid open, and it's not undisturbed. The animals are not undisturbed. So I think it's good to couple the information of behavioral tests with infrared thermography, but to use to monitor undisturbed animals, it's best to have them tagged and follow them using sensors that are.
Quick question, quick answer. Why are you considering all stress negative? Or Jose Antonio is asking.
I'm not considering all stress as negative. Of course, what stress is an integral part of life, but we have to mind this stress, and we have to also mind the effect, the impact on the research output. As you saw, there is no effect of habituation in terms of how animals react to handling, and also we showed that the animals will be in an aroused state, and even though this might not mean, if there is no distress, that there is a welfare problem, it will skew your results if you're not accounting for that.
Thank you, Nuno.